everyone. In this class, we will talk about G protein coupled receptor. This is the GPCR. Okay. So, what is GPCR? GPCR is a type of cell surface receptor. Okay, which is activated by the ligands such as either hormones, neurotransmitter, and other signaling molecules. Okay, so when this GPCR will activated, it initiated certain kind of signaling cascade through the process called the G protein coupled receptor signaling. Okay, so what is the ligand for this type of receptor? The ligand will be either hormones, okay, and neurotransmitter and other signaling molecules. Okay, then what is the structure of G protein coupled receptor? Here one receptor is there and one protein is there. Okay, so this receptor GPCR is a single peptide with a cytosolic region. Okay, a ligand binding extracellular region is there. Okay, and 7 hydrophobic transmembrane helix is there. Okay, so there is three parts. Okay, so one is the GPCR is a single peptide with a cytosolic region. Okay, this is the cytosolic region, and one ligand binding extracellular region is also there where the ligand is bind, and third one is the 7 hydrophobic transmembrane helix is there. Okay, so this is 7 transmembrane helical receptor it is. Okay, and in the cytosolic part there is one protein called as the G protein, which is made up of 3 subunit alpha, beta, gamma. Okay, so these two things complete the signaling pathway of GPCR. Okay, so this is the GPCR signaling. So what is happening in GPCR signaling? Firstly, the ligand will come and bind to the extracellular phase of the GPCR. Okay, and this ligand molecule nature is what? It is hydrophilic in nature. Okay, it is hydrophilic in nature. Then once this ligand will bind to the receptor molecule, it will lead to certain kind of conformational changes. Okay, so once the receptor is conformational changes happen, so it leads to the activation of G protein, which is present in the cytosolic part of the receptor. Okay, so this G protein in the normal condition, it is bind with the GDP. Okay, once the ligand is bind to this receptor, it changes the GDP to GTP. Okay, through guanine exchange nucleotide factor. Okay, and in step 2, G alpha subunit exchange GDP for GTP. Okay, so now this when this GTP is bind to this alpha subunit, it gets activated. Okay, and once it gets activated, it go to the adenylate cyclase. Okay, and it go to the adenylate cyclase and activate this adenylate cyclase, or you can say this is the effector molecule. This is the effector molecule. Okay, so this is the effector molecule and this is the hydrophilic ligand. So now what is happened? GTP bind to this alpha subunit of the G protein and activate it, and this activated complex go and again activated this adenylate cyclase. Okay, so once this adenylate cyclase will activate, it convert the cytosolic ATP to cyclic AMP. Okay, and this cyclic AMP convert ATP to cyclic AMP. Okay, so this cyclic AMP will what will happen? Elevated cyclic AMP level, then it activate the protein kinase A. Okay, so in the cytoplasm there is inactivate protein kinase A. Okay, there is two in protein kinase A, there is two subunit. One is the regulatory subunit, another one is the catalytic subunit. Okay, so this cyclic AMP will go and bind to the catalytic subunit of the protein kinase A and activate this inactivated protein kinase A. Okay, so once this activated protein kinase A, inactivated protein kinase A activated, it goes to the nucleus. Okay, and this will phosphorylate several enzymes and transcription factor downstream. Okay, which is example is cyclic AMP response element binding protein. Okay, this is the cyclic AMP response element binding protein causing gene expression that mediated cell growth and differentiation. Okay, it is mediated the cell growth and differentiation. So what is happening here? Firstly, the ligand will bind to the receptor. Okay, here the ligand will bind to the receptor. Then in the second step, through guanine exchange nucleotide factor GEF, which is called as. Okay, so what it will do is it will convert GDP to GTP. Okay, and here GTP to GDP is converted by GAP. Okay, guanine GTP is activating protein. Okay, so this is the GTP is activating protein which convert GTP to GDP. Okay, so guanine exchange factor will convert GDP to GTP, and in that case the alpha subunit will separate from this tri complex. Okay, and with GTP it will go to the adenylate cyclase and activate this adenylate cyclase. Once this adenylate cyclase will activate, it convert ATP to cyclic AMP. Okay, and because conversion of ATP to cyclic AMP in the cytoplasm, cyclic AMP concentration will increase. Okay, and this increased level of cyclic AMP will go and activate the inactivated protein kinase A. Okay, so protein kinase A is having two subunit. One is the regulatory subunit, another one is the cytosolic subunit. Okay, it is the homodimer, and after activating it, this protein kinase A will go to the nucleus and it bind to several response element binding proteins and causing the gene expression and cell mediated differentiation. Okay, so this is the GPCR signaling. Okay, and this GPCR signaling can lead to other kind of secondary messenger activation. Okay, like suppose this is this ligand will bind to the GPCR. Okay, and this ligand is activated the G alpha subunit of G protein. Okay, and once this G alpha subunit activated, it goes and activated the phospholipase C. Okay, in the previous case it was the adenylate cyclase, now it is the phospholipase C. Okay, so this type of means which kind of effector molecule will activate it, this is depend on the which kind of ligand is binding to here. Okay, so it is activating the phospholipase C. Once the phospholipase C is activating, it can secrete two type of secondary messenger. One is the DAG, that is the diacylglycerol, and third one is the inositol 3-phosphate. Okay, this one and this one. Okay, then this diacylglycerol will go and bind to the PKC, protein kinase C. Okay, and this will lead to the phosphorylation of substrate. Whereas this IP3 what it will do is it will go to the endoplasmic reticulum where the calcium ion channel is present. Okay, this is the calcium ion channel. It will bind here and activate this calcium ion channel, and it will lead to the secretion of calcium ion from inside to outside. Okay, and once this calcium will release outside or cytoplasm of the cell, it will go and bind to the PKC, protein kinase C, and this PKC will also bind to the DAG molecule, and it will lead to the phosphorylation of substrate. Okay, so these two type of uh, secondary messenger can be activated. Okay, through the G protein coupled receptor when the affected phospholipase C is the effector molecule. Okay, So this is the summary of GPCR. Okay, so here what is the summary? So here the neurotransmitter, both are the neurotransmitter as a ligand. Okay, non-epinephrine or glutamate. Okay, so this is the inhibitory molecule, I can say. But here sorry, so neurotransmitter, both is the neurotransmitter. Okay, non-epinephrine and glutamate. Okay, so if non-epinephrine is bind to the GPCR, then it activate the alpha subunit of GS alpha subunit, that is the stimulatory alpha subunit. Okay, and after activating this alpha subunit, it will activate the adenylate cyclase, which is the effector protein in this case, and then it will release the it convert the ATP to cyclic AMP. Okay, and because increase in the cyclic AMP in the cytosol, it will lead to the activation of protein kinase A. Okay, so protein kinase A will also increase protein phosphorylation. Okay, so this is the later effector. This is the primary messenger, and this is the secondary messenger here. Okay, whereas when the glutamate is bind to the GPCR, then it will lead to the activation of phospholipase C, which is here act as the effector molecule. Okay, then it can lead to two type of signaling. First one is the diacylglycerol, and the second one is the inositol 3 phosphate. Okay, and this DAG will directly go and bind to the protein kinase C and activate increase the protein phosphorylation and activate calcium binding proteins. Okay, and when this IP3 will produce, then it will go to the endoplasmic reticulum, and there it activate the calcium ion channel and release the calcium ion, and this calcium ion will go and bind to the protein kinase C. Okay, and it lead to the protein phosphorylation and calcium binding proteins. Okay, so this is the GPCR effector pathways. Now when means GPCR that regulate adenyl cyclase. Okay, how it regulate? So suppose this is the stimulatory hormone. Okay, stimulatory hormone is epinephrine, glucagon, ACTH. Okay, so this is the exterior part and this is the cytosolic part. So ligand will come and bind here. Okay, stimulatory hormone will bind here, so it will give the signal of activation of the G protein. Okay, so in that case the GDP will convert to GTP. Okay, and this alpha subunit will go and bind to the adenylate cyclase, and it will activate the adenylate cyclase, and it can produce the cyclic AMP from ATP. Okay, but ATP to cyclic AMP it will convert. Okay, but in case of suppose cyclic AMP concentration will increase in the cytosol, then what will happen is inhibitory hormone will go, that is the PG1 and adenosine. Okay, it is the inhibitory hormones, they will come and bind to the receptor. Okay, GPCR receptor, and then it transduce the signal of inhibition. Okay, so in that case alpha inhibitory subunit will activate. Okay, and in that case it can't convert the GDP to GTP. Okay, it will remain with the GDP. Okay, and here we can see the GDP, it will remain with the GDP. Now the beta gamma complex will go and inhibit the adenylate cyclase, so that it can't convert ATP to cyclic AMP. Okay, so to maintain the cyclic AMP level, inhibitory hormone will come. Okay, it will bind to the receptor and give the signal that the cyclic AMP should not produce. Okay, so in that case it will activate the inhibitory alpha subunit. Okay, how it will activate? It will remain the GDP to GDP bound form. Okay, and then this beta gamma complex will activate and go to bind with adenylate cyclase, so that it can convert ATP to cyclic AMP. Okay, then related to that, one question has been asked, choose the correct signal pathway. Okay, signal transduction pathway. Here it is saying hormone, 7 transmembrane receptor, G protein, cyclic AMP, then PK, okay, or protein kinase A. Hormone, G protein, 7 transmembrane receptors, cyclic AMP, protein kinase A. Then again hormone, 7 transmembrane receptor, G protein, protein kinase A, cyclic AMP. Then hormone, 7 transmembrane receptors, cyclic AMP, G protein or PK. Okay, so what is the correct sequence? So uh, correct sequence will be option A. How? Like hormone will work as a ligand here. Okay, so firstly ligand will go and bind to the receptor in the first stage of uh, cell signaling. We have seen that the ligand should be bind to the receptor. Okay, so ligand will be your hormone and it bind to the 7 transmembrane receptor, that is the GPCR. Okay, it will go and bind to the GPCR. Okay, then these three option is having hormone and 7 transmembrane region. Okay, option A, C and D. Okay, now you are having then after binding with the GPCR receptor, it leads to the activation of G protein. Okay, so in A and C is having G protein. Okay, after the transmembrane receptor, G protein in both the option is present. Okay, then once the G protein is get activated, it convert the ATP to cyclic AMP. Okay, so the correct option will be option A, because here protein kinase A comes first. Okay, which is not the correct sequence. Firstly the G protein will convert ATP to cyclic AMP. Okay, and once this cyclic AMP concentration increase in the cytosol, it lead to the protein kinase A activation. Okay, and this protein kinase A is having 2 catalytic subunit and 2 regulatory subunit. Okay, so in the catalytic subunit, this cyclic AMP will bind and separate from the regulatory subunit, and this will go to the nucleus. Okay, and in the nucleus it will bind with any kind of transcription factor. Okay, or CREB, which is the cyclic AMP response element binding protein. Okay, it will activate those kind of transcription factor and lead to the gene expression or cell proliferation. Okay, so this is the correct sequence. Option A will be your correct answer. Okay, so thank you all.